The overall goal of this immunostaining protocol is to detect protein expression in the aphid embryos. Critical conditions are identified that increase tissue permeability and decrease background staining, both of which are long-standing problems when staining embryonic tissues of aphids.
The main advantage of this technique is that it provides specific conditions for effective penetration of antibody and reduction of background staining, which usually are not described in the standard immunostaining protocol.
We first had this idea of this method when we were studying how germ cells are specified in the pea aphid, a writing insect model for genomics and developmental studies.
The laboratory strain of the Pea Aphid A. pisum was originally collected in central Taiwan and has been reared on host plants for more than 300 generations. To germinate, soak the seeds of the host plants in tap water for 3 to 5 days at room temperature. Refill the fresh water once per day. Grow 10 germinating seeds in a small pot with soil in the growth chamber under a photoperiod of 16 hours light, 8 hours dark at 20 degrees Celsius. About 10 days after growth begins, the height of the plants is usually more than 8 centimeters. Keep each pot of plants within a 1-liter glass beaker and transfer adult aphids onto the plants using a paintbrush. Then, seal the beaker with an air-permeable cover such as gauze mesh to prevent aphids from escaping. Incubate the aphids in a growth chamber under a photoperiod of 16 hours light, 8 hours dark at 20 degrees Celsius.